In this video, I'm going to talk a little bit more about DNA structure, some DNA vocabulary, and also talk about how this double helix will get squeezed inside of the nucleus, because it's going to take a little bit of folding and bending. So some terms that we've already been using, but I want to just take a moment and formally define, are chromosomes, genes, and DNA. So chromosomes are discrete units. That means like an actual physical unit of genetic material. And so to give you an example, humans have 46 chromosomes of these genetic units in our cells. So there are 46 actual physical objects inside of our cells that contain our genetic material. Another term that we'll see over the next couple of chapters is chromatin. Chromatin describes the DNA and protein complex, and it's more of like a chemical description. It's saying that this chemical is both made of DNA and protein, and chromatin can collectively refer to all of the chromosomes. So I could just say there's chromatin in the nucleus, and you would know that I meant there's 46 chromosomes in the nucleus. So that's how chromatin comes in. Genes are units of DNA within the chromosome. So if I imagine this is my chromosome, there's distinct pieces within the chromosome called genes. So this might be gene X, this might be gene Y, and these correlate to specific functions. So gene X might be the information that the cell uses to make enzyme X. This might be the information the cell uses to make protein Y. So genes are functional units that carry specific information. In eukaryotes, as I mentioned earlier, our DNA is linear. So again, there's a sidedness, one side, another side. Prokaryotes have circular DNA, so there's not a sidedness to it. And it's, of course, the double helix, this double-stranded DNA within that chromosome. So even though I drew it like this, there's really two strands in here. And same thing with this one. There's two strands of DNA here. So don't forget that whenever we're talking about chromosomes, we're talking about two strands. So the DNA in the form of the double helix, these two strands, is going to have to be folded and packed up to fit inside of the nucleus of the cell. So the reason why is that a chromosome is hundreds of millions of base pairs long, and if I imagined it stretched out, it would be a meter or 3 feet. So I'm taking 3 feet of DNA, and sure, it's really small as far as like you couldn't see it because it's microscopic, but it's super long. So I need to take that really long molecule and wrap it up and fold it down into something that will fit inside of that microscopic nucleus. So you start with your double helix. I'm not doing the double part in here just for simplicity, but you've got your double helix. And the first thing that's going to happen is that is going to get wrapped around proteins called histones. So the histones are in the center here. And notice that made it a little bit shorter because I have the loops in it. Then what's going to happen is the loops are going to alternate. So now I have the loops, and this is called a zigzag. The loops are one up, one down, one up, one down. And so again, that's making it a little bit shorter. And then the next piece of that is those are going to get, so this is my up and down loops. They get turned into loops as well. So kind of bunched. So we're going to go through these so you won't have to just rely on my slightly bad drawings. So the first level of compaction is the nucleosome. So again, there's my double helix, and the double helix is going to get, the double helix is going to get wrapped around protein. So inside of here is protein, and those proteins are called histones. So again, that's the protein. And the DNA wrapped around the histones is called a nucleosome. So the nucleosome is the DNA wrapped around the histone. So each one of these represents a nucleosome. And this is going to be held together because histones are positive. DNA has an overall negative charge, mainly because of the phosphate group. If you go back and look at the chemistry of a phosphate group, you'll see that it has a bunch of negative charges on it. So that's going to make DNA be negative, and so it'll therefore be attracted to the positive charge on the histone. That'll hold the whole thing together. And again, by wrapping like that, it shortens the length of the DNA. The next step is for the nucleosomes to zigzag. So here's one nucleosome, here's another, and it's making a zigzag. The zigzag will, by putting those up and down, makes it shorter. Imagine a piece of paper. So I have this piece of paper here for you, and as you probably know, it's 11 inches long, but if I fold it accordion style, so basically make zigzags in it, which that's what I'm doing off to the side here. I'm taking the paper and going up and down with it. There's my zigzag. That's not 11 inches long anymore because I've taken some of the distance and put it into the zigzag. So that's, again, shortening the length of the DNA. This structure overall is called the 30 nanometer fiber. 30 nanometers is the distance. It's just the distance across. So the distance from here to here. So it's the nucleosome zigzag or the 30 nanometer fiber. The 30 nanometer fiber, so that's what this is. So it's not showing you the zigzag anymore, just because the figure would get crazy complicated if they did. But this is the 30 nanometer fiber, and it is now getting made into a big loop too. That's held down to the nuclear matrix. So inside of the nucleus, there's a matrix of proteins that are going to have the 30 nanometer fiber stuck to it. So it's like taking a string and holding it at the base and making a loop out of it. So the nuclear proteins hold it at the base and make this loop. And again, that's going to make it compact even more. So DNA can be either really tightly packed or a lot looser. So compaction of the DNA is not uniform. So when DNA is very tight, it's called heterochromatin. So this is tight DNA. If something's really tight, it can't be read. So when the DNA is in the heterochromatin form, it can't be read or used. So this is going to especially happen when the cell is dividing. So that's when you will usually see heterochromatin. In euchromatin is when the DNA is loose. So when the DNA is loose, it can be read and used by the cell. And this is what the majority of the DNA is like when the cell is not dividing. So most of the DNA is loose when it's not dividing. And so if you were to look at a nucleus, so this is the nucleus. So when the cell is not dividing, the DNA is more loose like that, and one of the first signs the cell is going to divide is that the DNA becomes very punctate and dark. The reason why it becomes so punctate and dark is because the DNA condenses. Oh, whoops. So just a reminder, this is the nucleus we're talking about. So very loose DNA, this is your euchromatin. Very tight DNA, this is your heterochromatin. So again, division is going to cause the DNA to become very tight and for it to not be used during division. It doesn't need it during division, it doesn't need to use the DNA during division because it's really focused on dividing. So this is a summary of DNA compaction and structure. You have your double helix, which is loose and stretched out. In order to fit it in the DNA, it's going to start being wrapped around histones. This forms a nucleosome. The nucleosomes then take on a zigzag shape, which condenses it even more, and this is called the 30 nanometer fiber. And then the 30 nanometer fiber will get organized into these radial loops. The radial loops can get folded in on themselves, and this would be euchromatin when it's kind of loose. And then if it gets even tighter, this is the heterochromatin that forms when the cell is getting ready to divide. So this is the sort of classic way that chromosomes are depicted. But again, you're only going to see this kind of chromosome during division. So you only see DNA that compact during division, and that's your first sign that the cells are going to divide. And we'll talk more about that in later chapters.